Have you ever been walking down the street and seen an adorable cottontail rabbit hop by? Have you ever wondered why their cute coats have that marvelous pattern to them? I mean, at a distance, they look kind of brown, but up close, you can see a more complex pattern to them, that each individual hair is a different color at the base than at the tip, making a more intricate pattern. Well, it's thanks to genetics. Today, we get to explore the genetics behind what makes our bunnies look the way they do. In this lesson, we aim to provide an introduction to rabbit genetics, a glossary of genetic terms, and a discussion of how genes work together to control coat color and patterning in bunnies. Brief disclaimer though, in this unit we will be focusing on the 7 known genes that have been confirmed to affect rabbit coat color. But there is still much unknown about the rabbit genome, and there are so many genes whose functions have not been identified or clarified yet. So we're just going to focus with what we know. So, before we can begin discussing genetics, what is genetics? Genetics is the branch of biology that focuses on the study of genes and heredity. The study of genetics is essential to understanding what to expect in the offspring of particular pairs of parents. Genetic information is stored in the DNA, and the composition of those genes plays a large role in determining what we see and can measure when we look at an individual bunny. So let's say we have this adorable little friend here that looks like this. This is actually one of my bunnies, little Tommy. Let's use our friend Tommy here to help us define some keywords and concepts that we find in the study of genetics. Starting with phenotype. A phenotype is an observable or measurable trait of an individual, like Tommy's white fur or her long whiskers. Gene. A gene is the basic unit of heredity that consists of a DNA sequence at a specific location on the chromosome, such as the segment of DNA that codes for Tommy's fur color or whisker length. An allele is an alternative form of gene that differs in DNA sequence. It's synonymous with genetic variant, such as the allele that codes for white fur color specifically, or long whisker length specifically. And genotype, a list of alleles present at a particular gene of interest or in the individual organism as a whole, is the genotype. So when we're talking about genotypes, we can talk about someone being heterozygous or homozygous. Heterozygosity has to do with having 2 different alleles for a particular gene. For instance, Tommy has long whiskers. That's her phenotype. But the genotype for long whiskers could be she could have 1 allele for long whiskers, and then another allele that might not necessarily code for long whiskers. But if the long whisker allele is dominant to the other one, then the phenotype will still show long whiskers. And in this example, because she has 2 different alleles within her genotype, we would say she's heterozygous. Homozygosity refers to having 2 identical alleles for a particular gene. So if we're looking at Tommy's white fur coat, we could say if the reason she has white fur is because she has 2 alleles that both code for white fur, then she would be homozygous for that trait. Next, we have dominance, the expression of alleles of a single heterozygous gene where one allele has more effect on the phenotype than the other is dominance. Wild type. Wild type refers to the quote-unquote original allele or phenotype, meaning prior to domestication or before a mutation created a different allele. That is the phenotype we would see. And then we have the Punnett square, and you're going to be using this a good bit moving forward. A Punnett square is a grid that's used to determine the potential offspring of a specific mating pair. This form of prediction can be used to determine the genotype ratio of the offspring. A simple Punnett square is a 9-square grid. You can kind of see what I have here, but can increase to a much larger size depending on the pairings and the number of genes that are under consideration. So let's review the terminology using a real-life example, okay? In rabbits, the B gene controls the color base of the coat. The B gene has 2 alleles, big B and little b. The big B allele is considered the wild type allele, and it codes for black color. The little b allele codes for chocolate or brown color base. Assuming the rest of the rabbit's coat color genes indicate a solid dark color phenotype and that rabbit has genotype big B at the B gene, it will be black. A rabbit that is homozygous little b will be chocolate. A rabbit that is heterozygous big B little b will be black. Why? Because the big B allele is dominant over the recessive little b allele. So a rabbit with genotype big B little b will have the same coat color phenotype as a rabbit with the dominant homozygous genotype big B. So now let's discuss how genes work. Coat color in rabbits is controlled by several genes. Each coat color gene codes for a specific aspect, like color base, color density, pattern, extension of dark pigment, white spotting, or silvering. And just to clarify again, not all genes have been identified, and much is still unknown about the rabbit genome. There may even be as yet unidentified alleles for the known genes. We will learn more about specific coat color genes in our next discussion on coat color genetics, but first we're going to talk about additivity. Some genes have alleles that act in an additive fashion. That means that, unlike the big B gene example before, where the big B allele is dominant to the little b allele, a heterozygote phenotype would be about halfway between the two homozygote phenotypes. Genes with alleles in additive relationships play large roles in what we call quantitative traits, those that are quote-unquote measurable in numbers. An example of a quantitative trait is a rabbit's body weight at 10 weeks. The weight of a rabbit depends on its genetics and contributions from its environment, such as feed quality and how many litter meats it has. Let's use a made-up example and say gene W influences body weight. For a group of 10-week-old rabbits, let's assume all the environmental factors are exactly equal, all the same, and all of the other genes that contribute to body weight are also equal. Alleles big W and little w have an additive relationship. In this example, a rabbit with genotype little w weighs 1 ounce less than the average 10-week-old rabbit. A big W little w rabbit weighs the same as the average. And a big W rabbit weighs 1 ounce more than the average rabbit. All the alleles present in the genotypes can be observed in the phenotypes. They appear additively. With each additional W allele, the rabbit is 1 ounce heavier. We can view the inheritance of this relationship in a Punnett square, as you can see here. In this example, big W is high birth weight and little w is low birth weight. Offspring may end up as either high, low, or average, an intermediate heterozygote, due to partial dominance of the W genotype. So as you can see here, we crossed a heterozygous individual with another heterozygous individual and produced 2 homozygous individuals and 2 heterozygous individuals. However, the homozygous individual with 2 big W's, it weighs more, and the homozygous individual with the little w's weighs less than the average rabbit. So dominance. We've seen a bit about dominant recessive allele relationships and how they operate in this previous example with the B gene, right? That was an example of complete dominance, where the heterozygote, aka the big B, little b, has the same phenotype as the homozygous dominant allele genotype, big B, big B. We can fairly easily observe dominance in qualitative traits, which tend to be categorical, unlike quantitative traits, right? So coat color is a qualitative trait. Fur type, such as Angora, Rex, is also a qualitative trait. So the example with our W's are more for our quantitative traits where we have partial dominance, but in traits that are more qualitative, such as coat color, we usually more so see complete dominance. Imagine we have a mating pair of a male heterozygous black rabbit, so big B, little b, and a female homozygous black rabbit, so big B, big B. What will the offspring be? The genotypes of the offspring will be based off of the genotypes of both parents. During conception, the sperm will carry a random half of the male genome, and the egg will carry a random half of the female genome. This means that the sperm will only have one of the male's alleles, and the egg will only have one of the female's alleles. A Punnett square can be used to determine the proportions of each genotype expected in the offspring. In the cross of big B, little b, crossed with big B, there is a 50% chance that the offspring will be homozygous black, big B, and a 50% chance that the offspring will be heterozygous black, big B, little b. In this example, big B is black and little b codes for brown. And we have complete dominance of the big B over the little b. So if there's ever a pair of them both, we will only see the phenotype for the big B. If, however, we have a mating pair consisting of a female heterozygous black rabbit, big B, little b, and a heterozygous male rabbit, big B, little b, what will the expected offspring proportions be? In this cross, there is a 25% chance that the offspring will be homozygous black, so big B, 50% chance that the offspring will be heterozygous black, big B, little b, and 25% chance that the offspring will be homozygous chocolate, little b. And we can see this, right? I got this answer by making a Punnett square with both parents and filling it in. So we can see this right here. So that's another example of complete dominance with our B alleles. Let's now transition to epistasis. Epistasis is when the phenotypic expression of the genotype at one gene is dependent on the genotype at one or more other genes. It's a little bit like dominance, but instead of the interaction happening between alleles at one gene, the interaction is between specific genotypes of different genes. All right, so let's do this with an example. For example, the E gene can interact with the B gene to change the color base of the rabbit's coat. The E gene controls the extension of dark pigment in the coat color. Two alleles of the E gene are big E and little e. The big E allele codes for normal extension of dark pigment, meaning that the rabbit will appear as expected with the B genotype if the B genotype is present. The little e allele codes for no extension of dark pigment. When homozygous, the little e allele, so the genotype little e, only allows the production of yellow or red pigments over the black, so no black or brown pigments. So the genotype at the E gene can affect the expected outcome of the B gene. Genotype little e is epistatic to the B genotypes. A rabbit with a little e genotype would appear red, orange, or fawn, regardless of what is coded by the B gene. We will learn more how the coat color genes interact to affect phenotype in our next discussion. But moving forward, when we are considering two genes, the Punnett square gets bigger, with potentially more outcomes. So you can see that in the table that I've made below that shows the expected genotypic and phenotypic proportions of the offspring from a mating of an individual with little b, big E parent, and a big B, big E, little e parent. So setting up a two-trait Punnett square like this requires us to kind of recall back to our old days in algebra. The way that you set this up is for each genotype, you FOIL them, all right? If we remember FOIL from algebra, and you use the FOIL method to set up your Punnett square along the vertical and horizontal axes, okay? So for instance, genotype number 1 is this. And to FOIL this, we would do first to first, so that'd be little b plus big E. And then outside to outside, little b, little e. Inside to inside, little b, big E. And then last to last, little b, little e. All right? So you can go ahead and try to do that yourself for genotype number 2, the other parent, which is big B, big E, little e. So in this example, the B gene codes for fur color, right? The big B is black and the little b is brown, and the big B has complete dominance over the little b. And the E gene controls the extension of dark pigment in coat color, where the capital E, the big E, rabbits will appear as expected. And if there is a homozygous little e, then they will not necessarily appear as expected because those rabbits will always be red, orange, or fawn, not black nor brown. The coloring of offspring will depend on the interaction of the B and E genotypes. This is known as epistasis. Another example of this can be seen in the interaction between the B and D genes, where again, B codes for fur color, black versus brown, but the D gene controls the density of the fur color. So the coloring of offspring will depend on the interaction between the B and the D genotypes. Now, many students that take this course coincidentally seem to know a thing or two about cats. So I want to conclude our discussion here today with a brief disclaimer or reminder that coat color genetics in rabbits is actually quite unique and different from cats. The genetic basis for coat color in rabbits is different in that in rabbits, the gene responsible for determining coat color is located on autosomal chromosomes and not on the sex chromosomes, not on the X nor Y chromosomes. Therefore, the inheritance of coat color in rabbits is not tied to the sex of the individual. So unlike cats, male rabbits can indeed have a coat color with 3 colors, whereas in cats, generally, we only see that in cat individuals that have at least 2 X chromosomes, which are usually the female cats. Both male and female rabbits can exhibit coat colors with various patterns, including those involving 3 or more colors. The specific combination of colors in a rabbit's coat is determined by the interplay of multiple genes, each controlling different aspects of pigmentation and pattern formation, which we will touch on in our next discussion. So, in summary, while calico or tortoiseshell patterns are more commonly associated with female cats due to X-linked inheritance in cats, male rabbits can indeed have 3 colors in their coat patterns. The genetics of coat color in rabbits are influenced by multiple genes located on autosomal chromosomes.